Hi there everyone and welcome back to Higher Biology. The purpose of this video is to go through the 2019 Higher Biology paper, going through paper 1, which is the multiple choice paper. So throughout this video there's going to be 25 multiple choice questions going across all 3 units of Higher Biology, also a bit of problem solving as well. You can complete this in a couple of ways. I'll put a link to this paper just beneath the video. You can access this paper on the SQA website also. You can either go and do this yourself and then run through this while I go through the answers, or if you find it easier, I'll give you a chance to pause after I've introduced each question. Then you can attempt the question yourself, unpause, I'll tell you what the correct answer is, and then I'll talk about why that is the correct answer. If you do get it right, then well done, but one of the most important things is if you get your question incorrect, it's important to figure out where you went wrong, and hopefully my description will help you out with that. Sometimes it's just a case of misreading a question or not quite picking up on something that is asked within the stem of the question. Sometimes it might just be an area that you need to focus your revision on. So let's get started then with question 1, and I'll just scroll down here and introduce the question. So question 1 is about stem cells. It says embryonic stem cells can be used therapeutically to differentiate and replace any damaged or diseased tissue. And effectively the question is asking you why. So for the first question, I'll just remind you, you can pause this if you want. I'm going to give you the correct answer and then explain it. Okay, so the correct answer for 1 is D. They're able to do this because they are pluripotent. So hopefully remember, embryonic stem cells are pluripotent, unlike tissue stem cells which are multipotent, and all genes can be switched on. So the difference between D and B is it's important to remember that in pluripotent stem cells, all genes have the ability to be switched on, they can be switched on. It's not the case that they are just all switched on and they become anything. So question 1, the correct answer is D. For question 2, the table here shows the anticodon sequence on tRNA molecules and the specific amino acid that each carries. So in the table here you have a bunch of different amino acids and what their anticodon sequence on the tRNA is. And it says part of the amino acid sequence of a protein is shown in the diagram. So I'll give you a second to maybe attempt this question and again give you the answer. So the answer for question 2 is C. Now, how this question works is it's showing you the anticodon sequence. It then tells you the sequence of amino acids of the protein, so the proline, lysine, lysine, tyrosine, isoleucine. The question was asking you the triplets of DNA nucleotides that would code for this sequence. So you have to sort of work your way back here. So for example, if you use the table to figure out the anticodon sequence, you should find out the sequence of anticodons would be CCA, AAG, AAG, UAU, and AUU. So that's just from the table. You should be able to figure that out. Now, you should hopefully remember that what actually binds to anticodons, though, are codons. And again, we are still dealing with RNA, so we would not be using thymine, we'd be using uracil. So the complementary codons to this anticodon sequence would be GGU, UUC, UUC, AUA, UAU. So we've now worked out what these mRNA codons are going to be. But again, the question is asking you what the DNA nucleotides would be, so you need to go one step further back still. So the DNA sequence would be CCA, AAG, AAG, TAT, ATT. So again, this is what's quite difficult with this one here, is you're looking at the DNA molecules, and again, you're now bringing in the thymine molecules or bases instead of uracil. So again, be careful of that. A few things you have to do in this question. Question 3 is also quite a tricky one here. You're seeing a graph showing how temperature changes during repeated cycles of the polymerase chain reaction, or PCR. So in this question here, you're seeing the time in minutes on the bottom, going from 0 to 10, and you're seeing the temperature. So we're going through these stages of heating, cooling, and heating that take place during PCR. The question asks you, if there were 500 molecules of DNA at the start, predict how many copies there will be after 20 minutes. So the correct answer for question 3 is A, 16,000. Now, the way this works is if we first of all look at these cycles of PCR, as I said, you can see periods of heating, cooling, and heating, heating, cooling, and heating, and then finally at the end here, you have heating and then the start of a cooling period here. So across these 10 minutes, we're seeing 2.5 or 2.5 cycles of PCR taking place. Now, the question's asking you about 20 minutes, so obviously we'll double that. So it means across that 20 minutes, there is going to be 5 cycles of PCR. Now, if you remember, we're starting off here with 500 molecules of DNA, and for each cycle of PCR, we're going to be doubling these numbers of DNA molecules. So at this point, we just start to double for 5 cycles. So you'd start off with your original 500 molecules. After 1 cycle, you'd have 1,000. After 2 cycles, you'd have 2,000. After 3 cycles, 4,000. The 4th cycle would give you 8,000 molecules. And finally, that 5th cycle, hitting that 20-minute mark, would give you 16,000 copies of DNA, or 16,000 molecules of DNA. So a trickier question here, a bit of problem solving. You need to have a bit of knowledge of PCR, but also look at these different stages of PCR and how many fit into 20 minutes. For question 4, this is showing you the results of gel electrophoresis of a mother and a child and 4 potential fathers. So DNA from a mother, child and 4 men, A, B, C and D, in a paternity suit was analysed. The DNA was amplified using PCR and separated by gel electrophoresis. From the results shown in the diagram, identify the likely father of the child. So the correct answer for question 4 is D. It would be this man over here. So the first thing here is, if you look at the DNA of the child, and we're trying to obviously find the parents, we have the mother here, and you can see the first 2 DNA fragments match the mother. So these ones are the 50% of the DNA in this case that goes to the mother. So we're having to match these 2 fragments of DNA down here. So what you're doing now is it's got to match one of these 4 men. So I find it easier to use a ruler. If you go across, you're looking for one of the people, A, B, C or D, that's going to go and match this child. So for example here, you can see that B matches one of the fragments but doesn't match the other. Same with C, there's one of them, but D, both of these match together. So that means that D is the father of that child and matches DNA. For question 5, we have 3 examples of gene transfer. You have asexual reproduction by bacteria, sexual reproduction by mice, and plasmid transfer by bacteria. The question asks you, which of these examples illustrate horizontal gene transfer? So the correct answer for question 5 is B. It is only 3 that shows you horizontal gene transfer. So horizontal gene transfer takes place in bacteria. Try not to be confused with the diagram in number 1, the asexual reproduction, as both sexual and asexual reproduction are examples of vertical gene transfer where there is inheritance from one generation to the next. It's only in bacteria, that you would have this horizontal gene transfer taking place across the same generation. Question 6 is about mutations. So it says, sickle cell anemia is a human condition that affects hemoglobin, reducing the blood's ability to carry oxygen. It is caused by a mutation that changes one adenine base to thymine. This results in one amino acid in hemoglobin being changed. So the mutation that causes sickle cell anemia is, and you choose the mutation. So the correct answer for question 6 would be C, it's substitution. The reason for this is the question tells us that one base has been changed for another. So if it's only a single base that's been swapped or substituted for another base, that is going to be a substitution mutation. There's been nothing deleted, there's been nothing inserted into the sequence itself, and translocation is to do with chromosomes mutations rather than single gene. So 6 is C, substitution. For question 7 here, we find a bunch of different graphs here that show a subspecies of wren that's evolved in different island areas in Scotland. And the graphs show the averages of body length, wing length, and body mass for the wrens from the islands of St Kilda, Shetland, and Fair Isle. So there's a bunch of statements down here. I'll try and fit both of these onto the screen just now. So which of the following conclusions can be drawn from the data? Now, this one here is a lot of information being thrown at you. I'll just give you a second. You might want a bit longer to go through this one. Give us a pause and look at each of these statements. But you're trying to find out which of these statements is true. So the correct answer for this question is A. The Shetland and Fair Isle wrens have a greater average wing length and body mass than the St Kilda wrens. So you can see this if you have a look at the Shetland and Fair Isle wrens here. If you compare them in terms of their wing length and their body mass, so the middle one here and this one here, you can see that it is indeed higher. So the correct answer for this is A. If you go through the rest of the ones in B, you'll find that each one of them is untrue, so it's not the correct answer. For question 8, again, a bit of a longer question here, and I'll try and fit this all in, but it's talking about goldenrod gallflies lay eggs on the stems of several plant species. The newly hatched larvae then burrow into the plant, causing the growth of the mass of the plant, the tissue, the gall around them. The larvae live and feed inside the gall. So the size of this gall is affected by two different selection pressures, and this is where the speciation comes in. Larvae in smaller galls are more likely to be predated by wasps, whereas larvae in larger galls are more likely to be predated by birds. And the diagram here is asking you which diagram represents the type of selection affecting gall size. And the key here being for these diagrams is that the black line is the original population, and then the dashed line is the population after selection has taken place. So the correct answer for this question is B. It would be this diagram here. The reason for that is the question tells us that the larvae in smaller galls are more likely to be predated, but also the larger galls are more likely to be predated as well. So this means in terms of gall size, like looking at the graph here, it means that both extremes of the phenotype are predated, and therefore the middle, the average phenotype, is selected for. So this would be an example of stabilizing selection, where it's better to sort of be in the middle because that's being selected for, whereas the extremes on each end are what's basically what's bad, what's going to die off. Okay, so the small and the large gall size are going to be more heavily predated, so it's better to be in the middle here. And we can see from the original population, which was in the middle mostly anyway, you're starting to see the extremes even then encroaching in as well. So B is an example of stabilizing selection, and it shows what is happening in the question. For question 9, the diagram shows the divergence of lineages in the evolution of some primates. So you can see a bunch of different primates up top here, and the time, millions of years before the present, going from 0, present day, back to 80 million years. The question asks you, which row in the table identifies the time that the last common ancestor of vervet monkeys and humans existed, and the numbers of other species that shared this common ancestor. So I can't fit both of these on the screen at one point here really, but it's asking for the time, millions of years ago, where this last common ancestor of vervet monkeys and humans existed, and the number of other species that shared this common ancestor. Hopefully if you just look at the graph here, you'll hopefully be able to figure out the question without actually looking at the table before. So the correct answer for here is A. So 30 million years ago was the last ancestor of vervet monkeys. If you see the cursor here, you can see it. and humans. So if you trace these both back here, you get to this divergence point, which is 30 million years ago. The question always asks, though, also asks the number of other species that shared this common ancestor. So from this point onward, how many other species? There were 5. So the answer for this is A. Okay, 30 million years ago, and there are 5 common ancestors in this area here, apart from the humans and the vervet monkeys. Question 10 is going on to Unit 2 now. So the diagram represents the conversion of starch into maltose in this diagram here. Which row in the table identifies the type of reaction shown in the diagram and whether it requires or releases energy? So the correct answer for question 10 is B. So the type of reaction is catabolic. We can see the starch has been broken down into smaller, simpler molecules in maltose. That is a catabolic reaction, so large to small. But also when this reaction takes place, it releases energy. That's another part of catabolic reactions. So the correct answer is B. Anabolic reactions would be the other way around. That would be smaller, simpler molecules being built up into larger, more complex molecules, and that is also going to require energy rather than release it. For question 11, we see a respirometer, and the effective temperature on the metabolic rate of a snail was investigated using the respirometer shown. The experiment was carried out at 10 degrees Celsius, 15 degrees, 20 and 25, and at each temperature the tap was left open at the top here for 15 minutes, then closed, and readings were taken from the scale every 2 minutes. The question asks you identify how the dependent variable was measured. So the correct answer, so I'll move this down. The correct answer for question 11 is answer C. So the dependent variable here would be oxygen consumption. Now the reason for this is a bit of an experimental design question, is in this respirometer, this snail is going to be respiring. However, there is a solution here that's going to absorb carbon dioxide, so that shouldn't have any effect on the movement in this tube. As the snail is respiring, it's going to be taking in oxygen, and as the snail takes in oxygen, the coloured liquid is going to be... This means that oxygen consumption is then going to be measured on this scale, because you're measuring how far the coloured liquid is moving up because of how much oxygen has been pulled in from here. So the dependent variable in this case that's being measured is oxygen consumption. Question 12 says, which row in the table identifies the temperature monitoring centre and the location of an effector in thermoregulation in mammals? So the correct answer for question 12 would be C. So the temperature monitoring centre of the brain is the hypothalamus, and when there's a change in temperature, it's going to be picked up by effectors in the skin, and that's when thermoregulation is going to take place, where you're going to cool down yourself or warm yourself up. So 12 is C. For question 13, the Gila monster is a species of lizard that lives in North America. Its internal temperature is dependent on the environmental temperature. Question asks you which row in the table identifies the metabolic cost to and the range of ecological niches of the Gila monster. So the correct answer for question 13 is A. So the metabolic cost is going to be low and the range of ecological niches is going to be narrow. Now, in the question it tells you that if the internal temperature is dependent on the environmental temperature, then this animal is going to be a conformer. So conformers have a low metabolic cost as internal temperature is entirely dependent on the external temperature. But this also means, though, they can only occupy a narrow range of niches. So that comes into your regulators and your conformers, if you're not too sure about that part. Question 14 asks, which statement describes an event that occurs during the lag phase of microbial growth? So the correct answer for question 14 is B. Enzymes are induced. So in terms of your stages of microbial growth, you should remember that the first stage is the lag phase, where enzymes are induced, and this is before the rapid growth takes place, which is going to be in the log or exponential phase. And then afterwards, you're going to have your metabolites being produced during the stationary phase before you head into the death phase. So make sure you're comfortable with those. For question 15, using recombinant DNA technology, a culture of the bacterial species Micrococcus luteus was genetically modified with a plasmid containing a gene for a human protein. The protein was synthesized by the genetically modified bacteria, but it failed to fold correctly. Which of the following changes to this procedure may lead to a correctly folded protein being produced? The correct answer for 15 is B. You would use yeast cells rather than bacteria. So this is quite a standard recombinant DNA technology question. If bacterial cells produce incorrectly folded proteins, that's going to lead to an inactive protein. However, you might remember that yeast cells can be used to produce correctly folded proteins, as they are eukaryotic, so they have more complex machinery to produce these correctly folded proteins, which are essentially going to work. So any question like this, rather than using bacteria, you would use yeast cells. Question 16, again, I'll try and move this down so you can see all of it, says bacterial cells were exposed to disinfectant for 40 minutes. Every 2 minutes, a sample was taken and the number of viable cells were counted. The results are shown in this graph. It says calculate the percentage decrease in viable cells after being exposed to disinfectant for 6 minutes. Now again, there's some answers here, but if I just leave it here, you should be able to work out the percentage decrease anyway after 6 minutes. So the correct answer for 16 is A, 99.60. Now the reason for this, if you look at this graph on a logarithmic scale, we start with 1 million viable cells or living cells. However, after 6 minutes, if we count up, we have 4,000 viable cells that are still present, so quite a decrease. So what we do here, and be careful with the lines when you're having a look at your logarithmic scale, we're going to do a percentage change equation. So you find your difference between the two numbers, you divide it by original, and you times it by 100. So if you weren't too sure about that, you have 1 million take away 4,000, which is going to give you 996,000. You're going to divide that number by the original, which was 1 million. That's going to give you 0.996. And your final part for this percentage change is to times by 100, which would give you 99.6%, which, as I said, is answer A. Question 17 asks about field trials. It said a field trial was carried out to investigate the effect of applying different masses of phosphate fertiliser on grain yield of wheat cultivar. The wheat was grown in 3 fields. A mass of 50, 100 or 150 kilograms of phosphate fertiliser per hectare was applied. How could the design of the field trial be improved to minimise the effects of bias? So the correct answer for 17 is D. To minimise the effects of bias, you would divide each field and randomly apply different masses of phosphate fertiliser. Whenever you see a field trial question and it asks you about bias, you're going to go straight to randomising the samples. So the idea of randomising the samples is to eliminate bias, so there's not just one end of the field that has maybe more optimum conditions. It's got better light intensity, it's got better soil, it's got more fertiliser already there beforehand. So you scatter it out, you randomise them in order to eliminate bias in your sample. For question 18, fungi were cultured in a fermenter to produce an enzyme that was released into the culture medium. The graph shows the effect of oxygen concentration on the dry mass of the fungus and the concentration of enzyme in the medium after 48 hours growth. Question asks you the concentration of enzyme when the dry mass of fungus was 36 units per litre was, and again I'll try and move this up so you can see as much of it as possible. The correct answer for question 18 is B, 28 units per litre. So in this question you have to be really careful to use the correct scale and also the correct line. So with the fungus on the left-hand side here, when the fungus was we're also using the black dotted line, was 36, we find it here, that was at an oxygen concentration of 2. Now, if we then move up and match the enzyme line, which has the white dots to it, and we're now going to be using the scale on the right-hand side, we find that that is going across to 28 units per litre. So a couple of skills here. You have to look at the key, you have to make sure you use the right side. Okay, when it's top at fungus, use the left-hand side and the black line. When you're looking at the enzyme concentration, it's the right-hand side and the white dotted line as well. So answer B, 28 units per litre. 19 is asking about breeding programs. It says breeding programs can be used to increase starch levels in barley plants. This involves breeding closely related plants with high starch levels for several generations. This is carried out in order to what? So the correct answer here for 19 is D. It's to reduce the frequency of heterozygotes. So first of all, it tells you in the question here about breeding closely related plants. So breeding closely related plants, or inbreeding, is designed to reduce the frequency of heterozygotes. The idea of that is that you're left with organisms which are all true breeding or homozygous for that desired trait, such as in this instance, increased starch levels. So the idea of inbreeding is to reduce the frequency of heterozygotes. Answer D. 20, showing you a bar chart, shows the percentage decrease in yield of 4 crops as a result of the effects of weeds, fungi, and insect pests in crops A, B, C, or D. Predict which crop will show the greatest increase in yield if herbicides and insecticides are applied. So the answer for question 20 is D. Now the reason for this is if we have a look at this key up on the right hand side here, herbicides and insecticides will kill off the weed, which is the grey box, and also the insect pests as well, which is the black box. That means the only remaining pests after using the herbicides and insecticides would be fungi, which is shown in these white boxes here. In crop D, we can see that has the smallest decrease in yield due to fungi. So this crop would have the greatest increase in yield if the pesticides were applied in comparison to A, B, C. So for this one here, answer is D. Almost finished now. Question 21 shows a diagram showing the breeding of 2 parental pig breeds to produce the modern Duroc pig. You have a brown Duroc that's crossed with a Jersey Red, which produces a modern Duroc. The modern Duroc pig has increased quality and quantity of meat compared to both parental breeds. The development of the modern Duroc pig is an example of what? So the correct answer for this is B. It is crossbreeding. The breeding 2 pigs of different breeds would just be an example of crossbreeding. You're going to end up with a hybrid, in this case the modern Duroc. I can see there's a bit of hybrid vigour taking place here because this hybrid pig has increased quality and quantity of meat. It would not be inbreeding because it is not of the closely related pigs that are being bred generation after generation. This would be crossbreeding. 22 is looking into poor welfare. So it says a study into animal welfare in a zoo was carried out by observing the behaviour of some of the animals in their enclosures. The behaviours observed are shown in the table. Which 2 animals show signs of poor welfare? So the answer to question 22 is C, the lion and the white rhino. So pacing back and forward is a sign of poor welfare, so we should be getting concerned about that lion. The sun bear and the spider monkeys, though, are just showing entirely normal behaviour, as is the spotted hyena. Sleeping in the sun is not an indicator of poor welfare. However, that white rhino repeatedly chewing on bars, that is a sign of poor welfare, so that means that both the lion and the rhino are showing these signs. Answer C. And final page, everyone. So question 23 says, which of the following is not a behaviour of a worker honeybee? The correct answer for question 23 is A, producing eggs. So remember, workers are sterile. They do not produce eggs, they do not produce their own young. Their role in the hive is to gather pollen or food, they have to raise and feed the offspring, and they also have to defend the hive. So the only one of these behaviours that a worker honeybee would not do is A, producing eggs. In question 24, the statements describe examples of behaviour used by animals as protection from predators. Which of these are examples of social defence? The correct answer for question 24 is C, 2 and 3 only. So both 2 and 3 in terms of mackerels swimming in a large shoal or an individual meerkat looking out for predators while the other meerkats feed, they're all examples of a social group working together for defence. However, statement 1 is just an individual defending itself from a threat. So that would not count as social defence. Only 2 and 3 answer C. And your final question is about aspects of biodiversity. So the list describes some aspects of biodiversity, the number and frequency of different alleles in a species, the proportion of each species in an ecosystem, and the number of different species in an ecosystem. The question is asking you, which are components of species diversity? So the correct answer for 25 is D, 2 and 3 only. So number 1 is just about genetic diversity, that frequency of alleles, but the proportion and number of species relates to species diversity. You might remember as well the proportion, we would also term that relative abundance, and the number of species would be species richness, but they are both terms that we use to describe species diversity. So 25, the answer is D. Okay, so hopefully you've managed to get your way through these. If you went and attempted these all individually, it'd be good to look at your mark and, as I said at the start, more importantly, look at the areas that you got wrong. Hopefully describing the answers, I've been able to point out if you have got them wrong, where you have got wrong. And you'll find that a bit more helpful, and it's something to focus your revision on. The thing as well to really bear in mind is to take your time reading over these. You'll have noticed myself stumbling over so many of them as well. There's a lot of information being thrown at you. It will often confuse you. Take your time, go through it, and there are several questions there where the answer to the question was just embedded in the text of the question. So I hope you found this useful. I'm planning to do some more of these past paper questions videos. I know a few of you have asked for them. Hopefully you find this useful and you've done well with this. Best of luck for your studies, and I'll see you again for another past paper soon.